Hello everyone, in today's video we are going to tell you about how to make the DMEM Delbecos modified eagle medium and purify it. And the thing is 15 minutes of UV sterilization has been done, and we are wiping off all the surface of the this cabinet, and we will wipe it off all the utensils also with the alcohol. And now we are pouring the DMEM media into our workable Falcon tube and add it slowly. You can add with the serological pipette also, and this is also a method. We are making the 100 ml of the working media. So for the 100 working media, what we will need the 10% of FBS, for example 100 ml, that means 10% of FBS, and 1% of the antibiotic solution, and the 90% will be this DMEM media. So the thing is, for the 100 ml, what you have to do, you have to add 90 ml of media and 10 ml of fetal bovine serum, and 1 ml of antibiotic solution. So this is the instrument that we are using, is called as the Millipore filter. So in this Millipore filter, what you do, you will add the FBS, that will be the 10 ml, because we are making for the 100 ml of media. So you will add 10 ml of FBS, that is fetal bovine serum, into this instrument, and it is this Millipore filter. And after that, what you will do, you will take out the after this, you can set up according to you. If it is 1 ml, then you can set up it like that. And so you will take out the 1 ml of the antibiotic solution into the media. So this is penicillin and streptomycin. So because with this all the bacteria will be killed, and it won't work on the cancer cells, but it will work on the bacteria, so contamination will be less. And you have to attach the serological pipette into the serological tube, into the serological pipette, and take out the 90 ml of the its maximum amount. The maximum amount of the media can be taken is the 10 ml. So you will have to do it for 9 times, and you have to put it there in the filter. So 90 ml of this media will be given to this filter. So what we have done right now, we have added the 10% FBS, 1% antibiotic, that means 10 ml and 1 ml respectively, and we are adding the 90 ml of the media. And now attach the suction motor, so it will create the vacuum inside the bottom flask. Now the filtration process is completed, and now you can collect the bottom media into a new reagent bottle, and you can use as a working media for you. Thank you so much, guys.